So let's go ahead and start. This section, ready? This section kind of begins our study of what's called the central dogma of biology. You guys ever heard of that before? No? Well, I guess a dogma is kind of a way of living or a system or a knowledge or something like that. The central dogma of biology talks about this information flow that we're going to be looking at in the next couple of chapters. So in chapter 16, we're going to talk about DNA structure and replication and how information is stored in this. So information typically runs from DNA to RNA, and we'll specifically focus on messenger RNA and the information that it has. And then RNA is used to make proteins. So for most organisms, this is how information flows. We get the information from DNA and RNA to make proteins. What do proteins do in the cell? One word. What is it? Everything. They do everything, don't they? Pretty much everything. We've seen them be enzymes. We've seen them be transporters, so you were all correct. We see them in structure. We look at the cytoskeleton. The proteins are the things that are going to dictate pretty much all the traits and characteristics that you're going to see in the cell. So every cell has DNA. Ultimately, that's where the information comes and is used for all of its characteristics. When it wants to change any of its characteristics, it gets all that information from the DNA. And we saw how can cells change the proteins they have. Well, we saw some examples, right? When I talked about C3, C4, and CAM plants, they actually have cells that do different things. So if we look inside their cells, they have different proteins. So they're using the DNA differently. Doesn't mean they have different DNA, but they can use it differently for their different characteristics. So the first section, that's the dogma. If I ask you about the dogma, you say DNA, RNA, protein. That's the dogma of biology. There's processes within this, though. We're going to look at first DNA replication. So this is the first process. We're going to start with structure. I'm going to try to convince you of this information that's in this structure. And then on Monday, we'll start getting into the replication process. Replication is the process to copy the DNA. So if the DNA gives us all the instructions we need to survive and do well, do you want to give a good, faithful, high-fidelity copy of DNA to the next generation of cells? Yes, you don't want to give them a shoddy piece of DNA copy. That's not going to help the next generation adapt to new conditions or whatever. We're always trying to have the next generation be better off than the previous one. Okay? You'll learn that in ecology and evolution. Okay? So with DNA replication, that's going to be a highly regulated process to make sure it's done correctly. We can kind of treat these as pathways. So there's going to be a group of enzymes that are going to work together to do that. That's chapter 16. We're going to start with that. Chapter 17, this details what I have been calling gene expression. So gene expression, remember, is one way that a cell can control the proteins it has, whether it makes the protein or not. It's a pretty straightforward way to control them. We're going to look at how we make DNA to RNA. That's called transcription. And then going from RNA to proteins is called translation. So both of these make up gene expression. They're both done by independent enzymes. So we see each of these pathways are kind of independent from each other. So replication, transcription, and translation. And these are going to be tightly controlled as well. So you have to be careful of what you're doing, make sure you're making the proteins correctly. We're going to finally look at ribosomes, right? Ribosomes are what make proteins. So we're going to see how those enzymes extract the information from the DNA in order to make the proteins. Okay, so that's what this next section is all about. Looking at how DNA is made, this flow of information, how we make proteins, and all the enzymes that kind of go along with that. Ready to dive in? Yeah.
Let's do it.
Let's do it. Okay, so I'll probably draw this. Thank you for the enthusiasm. I like that. Especially on a Friday. Hard, summon. So let's jump in. We're going to look at DNA and DNA structure and the information that's in it. Okay, so I'm going to erase this. Okay, first off, DNA and RNA are called nucleic acids. So nucleic acids are going to be long polymers or chains of these nucleotides. So nucleotides are going to be the building block. By the way, this is the final macromolecule that we're looking at now. So we looked at sugars, and lipids, and proteins, and now we have the fourth and final macromolecule, which is DNA and RNA, these nucleic acids. Okay? So let's take a look at the structure of these things. And while we go through this, we'll kind of keep track of the differences between DNA and RNA. You want to be able to compare and contrast them. This is a typical nucleotide. It's a little bit scary, I guess, but you can kind of pick out some of the functional groups here. They might look familiar, like here, phosphate group. Seen that before. We have a sugar. So both DNA and RNA are made of types of ribose sugars. Okay, they do differ a little bit. And then we have this base that's also attached to the sugar. A couple things. One, you notice these numbers here: 1', 2', 3', 4', 5'. This is kind of a chemistry accounting for all the carbons that are on a molecule. If you get into nomenclature and Gen Chem or something like that, then you'll probably learn how these numbers are used so that you know what are basically bonded at each of those points, okay? So at the 1' carbon, then we see this nitrogenous base, kind of an ugly name, but why do they use this nitrogenous? What does that mean? Nitrogen. There's lots of nitrogen in here, okay? So that's why they call it. I'm just going to use the term base. We're going to see that there's different bases that can be attached to these nucleotides. At the 2' carbon, we have a difference here between RNA and DNA. In DNA, it's just a hydrogen, and in RNA, it's an OH group. What's that called again? Hydroxyl. Hydroxide's the ion. This close. That's okay. Don't be sorry. It's okay. It's a covalent bond here. So it's the hydroxyl group. Same thing here at the 3' is a hydroxyl. So that's our first difference in DNA and RNA. Keep track of it here. So we're just comparing DNA structure to RNA structure. So RNA is the regular ribose sugar, and DNA is deoxyribose. That's where you get the name from, DNA, deoxyribonucleic acid. You have to memorize that at some point. And then RNA is ribonucleic acid, basically. So if you look down here again at the 2', there's a difference between RNA and DNA. This does make a difference in the overall 3D structure of the molecule, but we won't really dwell on that. Okay. Now let's take a look at the bases that you can have. So here are the different nitrogenous bases. Where did I put my pointer? There's actually two different types. I have to move my little thing. Come here. There we go. So you have the pyrimidines, which are these single ring structures. That's cytosine, thymine, and uracil. Okay, you don't have to memorize the structures, but you want to get used to the names, okay? So pyrimidines are the smaller ones. Purines are these double ring structures, G and A. And we find there's differences in the ones that are used to build DNA and RNA. So in DNA, we find thymines, but in RNA, we find uracils. So that's another difference, is what kind of nucleotides are used to make these things. You guys have probably memorized this as well, right? The letters of DNA at some point. We have A, G, C, and T. So the enzymes that make DNA will use only those bases to do so. And then ribose is A, G, C, and U. So they use those particular nucleotides. We'll see more when we look at transcription and RNA structure. We'll see how the U's kind of come into play. They're really similar to thymines. Okay, you do want to remember which ones are pyrimidines and which ones are purines. I don't know a mnemonic for this one, but I always try to remember that pyrimidine is the longer word, but it's the smaller molecule. So it's kind of an inverse relationship. Purine is the shorter word, but they're the bigger molecules, okay? And so you want to remember that C, T, and U are pyrimidines, and A and G are purines. That'll be important later on. How are we doing? So you want to be able to recognize a nucleotide. You want to keep track of where things are bonded. So the base is at the 1'. You have a hydroxyl at the 3', and the phosphate's attached to which one? 5'. Okay, you want to keep track of that. And then the bases, depending on what nucleotide it is, you'll have different bases basically there, okay? Pretty straightforward. Okay, how are they put together? This is kind of interesting. When you connect two nucleotides, notice how they're connected. Here, this is, let's see, 1', 2', 3'. So this bond between two nucleotides, called a phosphodiester bond, fancy chemistry name, that is attached between the 3' carbon of one nucleotide and which one here? 1, 2, 3, 4, 5' carbon of the other one. Okay? So this is always the case when you're making a phosphodiester bond. It's going to be between the 3' OH group and the 5' phosphate group. So if you notice here, the 3' OH on one nucleotide and the 5' phosphate on that one. That's going to create the backbone of the structure. So if we make a long strand here, so this is a long polymer of DNA, then you'll see kind of a repeating thing. It'll go phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And that's the backbone of this molecule. A lot of carbons in here as well, right? So again, carbon plays a key role in forming the backbone of this structure. You guys see that? And again, this is going to be attached through an enzyme catalyzed reaction, right? So this is not just going to happen by itself. So we connect two nucleotides between the OH on one and the phosphate on another to create these phosphodiester bonds here, yeah? Very quiet, so I have no idea if I'm scaring you or if now this is DNA, so it's got to be much more interesting than metabolism, right?
Just maybe a dumb question about why is it kinked? Like, is there a significance to that?
Oh, I think, well, I mean, actually in solution, we're going to see that they kind of twist around each other in the helix structure. You just asked why this is kinked. I think this is just done for clarity, trying to show you the picture. So this doesn't necessarily look like what it's going to be. What about the bases? What are they doing? Anything interesting going on there? The bases, so I'm talking about those little ring structures, right? Are they part of the sugar phosphate backbone? Okay, what are the bases again? So these are the bases, right? The bases are attached to the 1'. So where are they on the polymer? They're the orange things, aren't they? The green things are the sugars. So it goes, this is the sugar phosphate backbone. Are the bases part of the backbone? No, we're not. Kind of hanging off to the side. This is important for the structure. Okay? Another thing to realize is there's a direction to this as well. So on one end, we have a 5' phosphate group. So we call that the 5' end that's sticking off. What about the other end over here? What's on the end? Yeah, 3' hydroxyl. So just like we read a book left to right, molecular biologists, we always read DNA 5' to 3' when we're looking at it, okay? By rule or convention. So there's a directionality here that's important. Kind of like the protein structure. There's a directionality to the protein structure. There's a directionality for DNA as well. How are we doing? You don't have to draw this picture, but you have to kind of understand how it's put together, how DNA is put together. Okay, the other difference that we see between DNA and RNA, I'll come back to this picture again, but here's another view of that, is that DNA is found in a double helix, typically, and RNA is really just a single strand. So you could tell this a double strand and a single strand. So the term strand is just referring to one polymer of the nucleic acid. So DNA typically is found to have two polymers intertwined with each other, whereas RNA only has one. So that's another difference that we're looking at here between DNA and RNA. So DNA is almost always double-stranded. And it's going to be found in this helix structure, which we're going to talk about some more. Whereas RNA is single-stranded typically. There are exceptions, but this is where we find them most of the time. This kind of structure. Okay, let's talk about this helix, because you guys have probably heard about the double helix before, haven't you? Watson and Crick. They used to make us read the book Double Helix in school. Probably not anymore. Okay, what I want to kind of go through is some of the information that was used to figure out the double helix structure, because Watson and Crick didn't come up with it on their own, kind of built on other things. And then we're going to focus on the significance of that double helix structure. Okay, so Watson and Crick. And this is like ancient history now, right? This was back in the 1950s, so a long time ago. Actually, it's kind of crazy. We've only known the structure of DNA for what, 70 years? It seems kind of short, doesn't it? So it's not that long ago in terms of science. Okay, Watson and Crick, they were the first ones to describe this double helix. Am I still on the screen? The double helix nature of DNA. But they used other people's information. So I kind of want to go through what they knew before they put this together. Of course, they won the Nobel Prize for doing this. Okay, they knew what the nucleotides look like. So previously, chemists had isolated DNA, and they were able to chemically describe what makes up those nucleotides. Okay? So that was published information. Watson and Crick knew what the nucleotides look like, along with all the bases. We're only talking about DNA right now, okay? So they knew what the bases were. They knew what it was made up of, what the molecule was made up of. So that was out there. Number two, there's another chemist named Chargaff. And Chargaff was an interesting scientist. He would go around and collect random samples of DNA from different places and isolate the nucleotides from that. So he knew what they looked like. And he counted them up. So what he discovered came to be known as Chargaff's rules, but applies to pretty much any double-stranded DNA. That the bases, some of the bases always occur in the same ratios. So whenever you looked at the amount of A's that were found in DNA, you saw the A's and T's, they were always around the same amount as each other. And then G's and C's were also the same. Within error, okay? It was never perfect, but they always seem to occur in the same ratios. So whatever you saw A, that was about the same as T. You didn't know why this was the case, but this was just kind of rule that for whatever reason, you'd always find these bases in the same ratios. Okay? Then the third piece of information that Watson and Crick had was from Rosalind Franklin. And this is kind of the controversial part of the discovery of the double helix. if you ever get bored, you should read The Double Helix by Watson. Okay, that's one side of the story. But read, I think it's called The Dark Lady of DNA. It's a biography of Rosalind Franklin and what she contributed to it. Because basically, the information that Rosalind Franklin got was shared by her boss to Watson and Crick without her permission. So I know that's kind of the immoral part of the story. And of course, Watson and Crick, and I always forget the boss's name, they all shared the Nobel Prize because Rosalind Franklin died before they gave her the Nobel Prize. But anyways, I know it's a sad story of how women are treated in our sciences. Not a good thing because women contribute a lot of good things to science. So anyway, it is what it is, right? We have to learn from it. Okay, who was Rosalind Franklin? Anybody know what kind of scientist she was? Yeah, crystallographer. She was an X-ray crystallographer, which is a crazy science. I have no idea how they even do it. But basically, you can make crystals of proteins or DNA, RNA, and you bombard these things with X-rays, hopefully in a secure room. You're not standing right next to it, okay? But somehow, from the diffraction pattern of those X-rays, you'll get some sort of design. You can look up the diffraction picture online. It looks like an X. That's all it looks like for DNA. But what she was able to figure out from that, which is a miracle in my opinion, is the width or information about the shape of the DNA molecule. Okay, so she was an X-ray crystallographer, and what her experiments told her was kind of about the width of the DNA molecule and also kind of the spacing between the different nucleotides within it. Okay, so she got, I don't know, what would you call it, physical characteristics or something of it? Yeah, something like that. So she got the width and kind of spacing of the nucleotides, which gave Watson and Crick important information on the overall molecule. This is typically how science works. People use a lot of previous information, usually when it's published, okay, to then extend the theories and things in science. So this is what Watson and Crick did. They used the information they had, whether they got it the right way or not. They used that. I always have to put the disclaimer on there, sorry. They used it to come up with what's called a base pairing. That's what they came up with. And that allowed them to publish their paper and win all their awards for doing it, okay? So they used this information here. They used the info to come up with what they called base pairing. Base pairing, okay? We're going to take a look at what this is, but this idea of base pairing, Watson and Crick base pairing, gave them the foundation they needed to come up with the double helix structure, okay? You guys got all that? Okay, we're going to look at more pictures now, okay? So you can see kind of the picture there of the base of the double helix, how they're wrapping it around. But base pairing is this idea right here. Watson and Crick noticed that when you put the nucleotides together, you can form some of these things that we don't like to talk about much anymore, right? What are they from chapter 2? What are the dotted lines? Hydrogen bonds. You can never leave chapter 2 alone, okay? Well, hydrogen bonds are a result of what? Partial charges, which come from where? Polar covalent bond. What are the polar covalent bonds you're supposed to memorize? OH, CO, NH. What do we see here, folks? Look, we've got NH groups, NH, CO, NH, CO, okay? So these hydrogen bonds allow the nucleotides to interact with each other. The other thing to notice here is that these pairs always occur between G and C and A and T. So this explains why Chargaff found these things in the same ratios, because they're always in the same base pairs with each other. GC pairs are able to form 3 hydrogen bonds each. Okay? Oh, by the way, this nitrogen here, you'll learn in chemistry, has a lone pair of electrons, which basically can interact and form a hydrogen bond as well. A's and T's, though, this is covered at the bottom. A's and T's only, they only make 2 hydrogen bonds, okay, not 3. So they're a little weaker. Okay, okay, well, why is this important? Well, it's important because it describes then how the nucleotides are bonded to each other in this base pair here. So if we see the DNA, we have 2 strands of DNA. So there's 2 polymers here. Each strand has the nucleotides sticking off here. And then what we see in the middle is the hydrogen bonds between the AT and GC pairs, okay? Okay, which ones were pyrimidines and which ones were purines? Yeah. G and A are the bigger molecule, so they're the smaller word. Which is what? Purines. They're purines. C and T are pyrimidines. Notice that base pairs are made up of 1 of each, aren't they? 1 purine, 1 pyrimidine in each pair. Okay. So this was really important because it gave you a uniform kind of width to the molecule. If you look at how wide it is just at the outermost point. If you have the same base pairs, then you always have the same distance in angstroms, you know, which is, I think it's like 10 to the 9th meters or something. You always have the same distance there. So that's what Rosalind Franklin found. She also figured out the spacing between these. So by looking at that X-ray diffraction, she could figure out how far apart they were from each other. Isn't that kind of cool? So using information about the size of the molecules, how the molecules form hydrogen bonds, that allowed Watson and Crick to figure out that these 2 strands of DNA could interact with each other. And they also made an interesting point. These aren't covalently bonded, right? And hydrogen bonds tend to be weaker. So what do you think is the significance here of that, of having hydrogen bonds instead of covalent bonds?
So you can break them apart.
Yeah. Yeah. So you could take a DNA molecule and heat it up. You could heat it up and break them apart. That's not a good strategy for cells. What do you think cells do? What do they use? Proteins. Yeah, proteins. So they'll have enzymes that come in here and break these apart to separate the 2 strands. But that's where the information is. The information is in these bases, in these base pairs, okay? So you have to be able to get into the inside. Okay, let's play around with this a little bit. We'll come back to the structure. How are we doing? Kind of cool? Interesting? Okay, let's just draw a DNA molecule, and I'm not going to draw out the sugar phosphate backbone. What I'm going to do is draw out the sequence of the bases as they're found in 1 strand, okay? I'm just going to make it up. So we'll do G, C, T. Okay, I'm just making something up. So using Watson-Crick base pairing, can you figure out the other strand? It's called, I always can never say it, it's the complementary strand, or the complement. Go ahead and try it out in your notes. We haven't done parent share a while. Why don't you show your partner once you get the other strand? So we give you 1 strand of DNA, you want to come up with the other strand of DNA. So much fun. Yes, sir.
So it's the reason that RNA is you and your cells instead of vinyl, because the binding doesn't read well with the,
Well, one of the theories, if you look at the structure, they only differ by. One of the theories is if you had uracil and TS, then the pairs wouldn't know if that was situs binding that lost. It's supposed to be uracil. So that's one of the theories. So you don't have uracil there. Yeah. My last one we were talking about. One of the theories is we don't have uracil in TNA. Because we look at uracil and thymine, we only get the one. So thymine were to lose the messenger, damage, then the parent enzyme wouldn't know whether it's thymine or uracil. So one of the theories if you look at how they continue to age, they still form the same way. Okay, so we're not doing Chargaff's rules here, right? We're doing Watson-Crick base pairing rules. That's something to keep in mind. Remember, Chargaff had no clue what a base pair was. Okay, just keep that in mind. Okay, what is it? So G's here, yeah, always binds with C, right? So you just follow the rules. G's and C's always go together, and then T's and A's, yeah? Pretty straightforward, isn't it? Yeah. Okay, what did I leave off? Yeah, the directionality. So if this is 5 prime, then this end better be 3 prime. But what about the other side? I didn't point that out to you. Yeah. The fancy word here is anti-parallel. So the 2 strands are anti-parallel. If you look at the 5 prime end on this side to 3, look at this side, 5 to 3, okay? So that's always true for DNA molecules. The strands are kind of going in different directions, aren't they? When you look with respect to the 5 to 3 prime direction. Which means as molecular biologists that we kind of read them differently, don't we? Yeah, because if this is 3 prime, this one's going to be 5 prime to 3 prime. If I wanted to look at the sequence or read the bottom, I would start on that side and go 5 to 3. Okay? So we would read these 2 strands differently depending on the strand we're actually looking at. And we can use either one. So when we get to this transcription, we can see that we can transcribe either strand. It just depends on the gene. Okay, come up with the proportion of A. So how much A is there? Go ahead and count them up and do it in your notes. Both strands. So when you're looking at double-stranded DNA, you look at both strands. When adding up all the nucleotides. Yep. So you can just give me a fraction. I don't know. It might be ugly because I didn't count them up. Part of math. Got it? How many A's are there? 8 out of how many? 28. 8 out of 28. What about T's? What? You're kidding me. They're the same? Well, yeah, they better be the same, right? What's the rule? I shocked you a little bit too much? Okay. 8 out of 28, okay? So A's and T's should be the same. Why? Because they're base paired together. They're base paired together. Chargaff, again, didn't know that, but that's what his rules were stating, okay? So if that's 16, that means this is 12 and 12, right? Physics file and whatnot. It's not 12. They have to add up to 100, don't they? Or at 28. 28, 28, 6. 6 and 6, is that right? 16 plus 12, I hope so. Then it'd be really embarrassing. Okay, this only works for double-stranded DNA. So if you looked at single-stranded DNA or RNA, then Chargaff's rules don't really apply. Okay, so this is only true for double-stranded DNA. So that's kind of neat, huh? Because if I just gave you one number, could you figure out all the rest? Yeah, that would be a test question, okay? Easy test question. You guys can figure that out. And if I give you one strand of DNA, can you figure out the other strand? Yeah, easy, isn't it? Very easy. Okay. Nice, easy question. Any questions on that? Okay, so let's talk a little bit more about the structure. Where is the information? I told you this. Yeah, it's not in the backbone, but it's in here in the middle, in those bases. And specifically what it is it's the sequence of the bases as you go along the DNA strand. So the DNA is made up of 4 different bases, but you can put them in lots of different sequences. So we just look at the letters. This doesn't correspond to this molecule. But this is kind of what we talk about when we talk about the DNA sequence. It's going to be the bases in order as you look at that DNA molecule. So what we're going to learn is how the cell gets that information out of the DNA and RNA and makes proteins from it. It really goes back to those letters. You won't have to draw DNA like this ever, but you do need to realize what the letters are referring to. Okay? And this idea of DNA sequence. That's what they always talk about when they sequence DNA. Anytime you get your DNA sequence. Anybody ever do that? You don't have to tell me, but they have those companies now, right? 23andMe or something like that, where you can actually get part of your DNA sequence. So it can be kind of interesting. Okay, later on we'll talk about our genes, not the genes we're wearing right now, but the genes are found in our DNA. And those genes then they give the information for making whatever proteins that we're going to make on the cell. And again, it's all based on the sequence of letters that's there, okay? So we'll come back to this when we talk about cell cycle, things like that. I think we start that after Thanksgiving. I don't remember. But when we talk about that, we'll talk about chromosomes and DNA structure, okay? Okay, any questions? No, we're not going to go any further. Expectations? Any questions on expectations? Yes, sir.
So, can you go over what is attached to each, like, carbon again?
Yeah. What's attached to each carbon? Sure. Yeah, the picture of the nucleotide? Yeah. So, yeah, and we're only concerned about the numbers on the sugar, so not the numbers on the base. The base is always attached to the 1' carbon of ribose. Remember, these all represent carbons here, the intersecting points. The difference between DNA and RNA is found at the 2' carbon. The 3' should always be a hydroxyl group, and that's important for the phosphodiester bonds. 4' we don't talk about it. 5' though, that's where the phosphate groups are attached. I think you have this picture, and that one doesn't really show it, but you have this picture as well. It doesn't show the numbers, but you can always count. In chemistry, you learn the rule is you always start on the right-hand carbon, right, from that oxygen. You guys doing nomenclature in chem? I have no idea when they do it. Gen Chem? Anybody taking Gen Chem? I don't know. You'll go through all the rules, especially if we get to OChem and have to name these difficult molecules, then you have to know where you're talking about. So yeah, you don't need to memorize or draw any of the bases, thankfully, but you do need to remember which ones are pyrimidines, which ones are purines, and how they are related to each other in those base pairing relationships, okay? So this picture here, how they're paired up. Okay, so on Monday, we will get into the importance of DNA and DNA replication.